Hello students, welcome to our channel. Today our topic is generation and conduction of nerve impulse or action potential. So let's see. At first we will see different phases of the action potential. The first one is polarized state. In this phase, the potential difference between the inside or outside of the axon is minus 70 millivolt. This is also called resting membrane potential. In this phase, the sodium potassium ATPase pump are activated, and through this pump 3 sodium ions get out of the axon and 2 potassium ions get inside the axon. Then comes the local potential. It is the phase after the stimulus is given. In this phase, the potential from minus 70 millivolt increases up to minus 55 millivolt. This minus 55 millivolt is also called the threshold potential. In this local potential phase, slow influx of sodium channel occur. Then comes the depolarization phase. In this phase, the potential difference goes from minus 55 millivolt to plus 40 millivolt. In this phase, there is a part from 0 millivolt to plus 40 millivolt. We called it the overshoot of action potential. In this phase, the voltage gated sodium channels opens. When the potential difference is minus 55 millivolt, the voltage gated sodium channels get opened. And due to this, rapid influx of sodium plus occurs, which increases the intracellular positive charge. Then comes the repolarization phase. In this phase, the potential difference comes down from plus 40 millivolt to minus 70 millivolt, which is the base potential. The mechanism in this phase is K plus efflux from the axon. Through voltage gated potassium channels, the potassium ions get out from inside axon to the outside. Then comes the hyperpolarization phase. In this phase, the potential difference between inside of the axon and outside of the axon become minus 70 millivolt to minus 90 millivolt. It occurs due to slow closure of potassium channels and influx of chlorine ion. After the repolarization, all the potassium channels cannot get closed simultaneously. So they close slowly. Due to this slow closure, the potassium efflux continues and the hyperpolarization state is seen. Now we will see these things in a graphical form. At first, the minus 70 millivolt, which is the resting membrane potential. At this situation, the stimulus is given and the curve starts to get up. In this phase, we have already seen that slow sodium plus influx occur. When the curve reaches the minus 55 millivolt, all the voltage gated sodium channels get open and rapid influx of sodium occurs. Due to this, a rapid surge is seen in the curve. It comes up to plus 40 millivolt. Now, at 40 millivolt, all the sodium channels get closed and the potassium channels get opened. Due to this, the potassium gets out from inside the axon to outside and the curve get down rapidly up to minus 70 millivolt. Now, at minus 70 millivolt, all the potassium channels are not able to close simultaneously. Due to this, more K plus efflux occur, and with it also the chlorine ion influx occur, which causes the potential to decrease up to minus 90 millivolt. After that, when the potential reaches up to minus 90 millivolt, the sodium potassium pump gets activated. The potassium channels also get closed and the chlorine influx also stop. So the curve then rises again up to minus 70 millivolt, which is the resting state. Now we will see what is the refractory period. There are two types of refractory period. One is absolute refractory period and the other one is relative refractory period. The absolute refractory period is from the threshold point to 1/3 of the repolarization state. In this period, if any other stimulus is given, no response is seen in the neuron. The relative refractory period is from the 1/3 of repolarization to end of the action potential. In this period, If the second stimulus is given at maximum strength, then response is seen. Now, in these pictures, we have shown the different phases of action potential, and we have also shown the conduction of action potential. At first, you can see the polarized state. Here you can see the sodium potassium ATPase pump is active. This is the sodium potassium ATPase pump. It causes efflux of 3 sodium ion and influx of 2 potassium ion, which helps to maintain the negativity inside the cytosol and the positive charge in the extracellular fluid. Then come the depolarization phase. In this phase, you can see the sodium channel, which are the voltage gated sodium channels, get opened and huge amount of sodium gets inside the axon, and the charge changes from negative to positive inside the axon. You can see that, and the outside charge become negative. So this phase is called depolarization. Then comes the repolarization phase. In this phase, you can see the sodium channels are closed in this area, and the voltage gated potassium channels are open. Due to this, huge amount of potassium get efflux from the axon, which cause again the change in charge inside from positive to negative, and the outside charge change from negative to positive. Here you can see another thing, that in this photo, this part is depolarized, and after that, in this photo, this part gets repolarized and the depolarization shifts forward. So in this way, the action potential conduction occurs. It is very important to remind that in myelinated neurons, the action potential generates at the nodes only, where the myelin sheath is not present. So the nerve impulse travels by jumping from one node to another node. There is a law called all or none law. It means a neuron either responds completely to a stimulus with a maximum action potential, or it does not respond to it at all. So this is all for today. In the next video, we will come with another interesting topic.